Okay. Good morning, everybody. How are you all doing? So, this is Mohan Kundu, and I taught this course Essentials of Biomolecules, which mostly comprised of nucleic acids, peptides, and to some extent we have also covered carbohydrates. And as you have gone through some of the lectures, we have more to come. Most of these topics that I have covered includes chemistry, organic chemistry, and a little bit of biology of the biological macromolecules. How DNA is formed, how DNA is replicated inside the cells, how we can manipulate in the laboratory, how we can develop organic chemistry techniques, biochemical techniques to manipulate different aspects, different properties of the nucleic acids, how we can synthesize new kinds of nucleic acids which are not natural, that can be used for so many purposes, including the pharmaceuticals, including the drugs. We have also seen how peptides are developed, how peptides can be synthesized in the laboratory using the solid phase peptide synthesis, how different chemistries are involved, different reactions are involved, how there are so many variations in the nature that the functional groups of the amino acids are varying, and you can do really a lot of chemistry to use those amino acids or to use those properties. And of course, we have also seen to some extent the carbohydrates also, the natural carbohydrates and carbohydrate containing drug molecules. So, overall those were the structure of the course that includes a lot of chemistry, chemical synthesis, chemical reactions, and biological techniques. So, I will be happy to take some of your questions or queries or doubts that you have on the lectures that we have gone through so far. How many people are there? 3. Oh, okay. So, did you like the lectures, the initial ones? Are the lectures helpful in understanding some basics of the chemical biology? जो जो क्वेश्चंस पूछा था वो लोग है लाइव में जनरली आते हैं लोग आते हैं। मिनट्स के बाद बंद कर सकते हैं ना 15। So we have got a question that states that doesn't it require enzyme to break and replicate, where enzymes also present in prebiotic chem. So, prebiotic chemistry. To answer the last part, so when we talk about prebiotic chemistry, that entirely means there were no live things during that time. There were only the basic elements or the basic molecules, very basic molecules, like we have stated, like ammonia, like methane, like carbon dioxide, and maybe a little bit complicated that came a little later, but obviously no large molecules and certainly not the biological macromolecules. So, in prebiotic chemistry there were no enzyme involved, enzyme present, there were no proteins available. So, whatever happened, whatever the molecules that had been synthesized in prebiotic earth, those were basically formed over a long period of time, millions of years of times, and that tells, when everything, all the small molecules are present in a soup, in a primordial soup that we call, or in a broth that was all molten state, and then they stay for millions of years, then there is of course a probability that some molecules would come in close contact to each other, and under certain conditions, under the given condition that were present during the prebiotic earth, they might have reacted to produce a little bit larger size of molecules, and some of those larger molecules happened to be the basic units of the biomolecules, such as the amino acids, small amino acids, such as the nucleobases, such as sugars. So, different entities might have been evolved over a very long period of time, and then they have again clubbed together to synthesize a little bit even larger molecule, and that is how the first live thing that is kind of predicted or kind of assumed to be the kind of amoeba type of cells that have been synthesized. So, when you talk about the enzymes or the proteins, certainly they were not present at the prebiotic earth. They came much later, much later. And the first part of it, I am not very clear of what you wanted to ask. Doesn't it require enzyme to break and replicate? A lot of enzymes, of course, are required for replication of DNA. I don't know if that is what you wanted to mean. So, when you talk about replication, replication means we usually mean that it's a multiplication of DNA, which of course requires a lot of enzymes that we have seen stepwise. And at first, obviously, the double stranded DNA needs to break into two individual strands, not fully, that forms a replication fork that I have talked about, partial opening of the DNA double strand, and that is done through an enzyme, of course, that we called the helicase, enzyme helicase, that will break the double stranded DNA partially into two single stranded DNA. So, enzyme helicase is involved to break the initial of the DNA double strand, and then that is being stabilized by single stranded binding proteins, followed by the other enzymes. But I think if you wanted to ask something else, can you state it little bit clear? What is the second question? Sir, ITA, not full question. What is this? Okay. Actually, I am asking, after that nothing else? Okay. How did first single cell organism knew how to replicate? Okay. All right. So, first single cell is presumed to be kind of amoeba, where there are no DNA. So, that is made up of mostly RNA. The organisms mostly are based on RNA, and a large part of scientists, biologists, evolutionists, they believe that the biomolecules, first biomolecule that had been synthesized on the primordial earth or prebiotic earth were RNA, not DNA, not proteins. So, when it came to RNA, obviously that did not replicate as such. It only that merely many of the RNA have been synthesized, and so that the many organisms were there. And after that, all the details I also do not know, because I am not really an expert in the biology in the of evolution. So, that is the basics that I know. Thank you. Thank you. If that helps, yes, thank you. Any more queries are there? Hello all, do you have any questions? If there is any doubt in the lectures we have covered so far, you can easily ask. All right. So, it looks like there is no more questions there. So, if you have any questions, please fire away quickly. Otherwise, I think we can conclude the live session, because if there is no other questions, then we can conclude the live sessions. All right. So, thank you so much for attending, those who are watching, and I wish you all the best for future. Hopefully that the lectures will help you gain some knowledge in chemical biology as well as biological chemistry. Super. So, thank you so much for watching. Take care.